Hello, Gen Bio. Dr. Fry here, and this week we're going to talk about animal development from the point where an unfertilized egg becomes fertilized, divides through successive mitotic divisions, and then develops into a larval form, and then from that point to juvenile and even an adult. Okay, so let's start with a few directional terms. So just when we look at the slides in lab this week, I might use words like dorsal, ventral, rostral, caudal, and I want you to know what those mean. So there's this very classy lady who is going to help me to demonstrate these terms. So dorsal is towards the back. Okay, that's always going to be towards the back. It can be on a two-legged organism such as ourselves, or in a four-legged animal, where the back would be pointing up instead of behind you. But that towards the back is always going to be dorsal. Okay, and then towards the chest is going to be ventral. Same concept. It's going to be towards the front of the organism, towards the face, chest area. Okay, so that's ventral. Rostral means towards the nose. Caudal means towards the tail. Midline is going to be median, and lateral can be either direction from out from the midline. Okay, and then anterior towards the head, posterior towards the backside. Okay. All right, let's move to our next slide. So to look at animal development, we have to look at various stages, okay? And all of this begins, of course, with fertilization, where you have your haploid sperm meeting your haploid egg. And this will, of course, produce a diploid zygote. So that, as we discussed previously, will restore the full number of chromosomes, and then you have a diploid organism, okay? This is followed by successive divisions through mitosis, but there's not a lot of cell growth, okay? The cells, that one single fertilized cell that occurs when sperm meets egg is going to divide into 2, into 4, into 8, and so forth. But this period that we call cleavage does not have a lot of cell growth. It's just a lot of rapid cell division. So that 8-cell stage is going to be roughly the same size as the fertilized egg because there's not a lot of growth happening. It's just a lot of division. Okay. And then following the period of cleavage is gastrulation, where you've got a movement of all of those new cells. So they're gonna shift. Sometimes they're going to overlap and form layers. And this rearrangement of cells forms the 3 germ layers. So you'll hear me use the terms ectoderm, mesoderm, and endoderm to refer to the 3 germ layers of tissue, which will later develop into the parts that we recognize as the fully formed, differentiated parts of the body. Okay. Following this stage, gastrulation, is organogenesis and neurulation. And this is where those layers, those germ layers, will take on specialized functions. So you've got differentiation of your cells, more rearrangement, more movement of the cells, and this will form the different organ systems and the nervous system. Okay, so organ systems is specifically referred to as organogenesis, and the development of the nervous system is called neurulation. Okay, and then following this point, we have a development from a larval form to a reproductively capable adult. Okay, so this can look different in different organisms. So the organisms we're going to look at in lab today are model organisms. They're easier for us to examine microscopically, easier to come by than, say, more advanced organisms such as a monkey or a human, so forth. There's a lot of ethical implications that come with looking at those, as opposed to the amphibian, which we'll look at, and the sea star. So we have access to these, and we have lots of slides that you'll look at to look at the development in these organisms. So one point I'll make about metamorphosis for these organisms is that the starfish, as a larval organism, as in its larval stage, it actually exhibits bilateral symmetry. So bilateral symmetry is if you cut something down the midline and you look at the both sides of it, it's going to be symmetric left to right. Okay, there's symmetry, body symmetry. This is not what we think of when we think of the mature adult starfish, the sea star. Typically, this, as an adult, is an example of radial symmetry, where you have those often 5 arms that come out from the center. And so that's an example of radial symmetry, but as a larval form, the starfish actually has bilateral symmetry. So that's a metamorphosis that the sea star goes through during this period of development for it. The tadpole then is obviously the juvenile form of the frog, which has a different appearance. It has a tail, for instance. It has different habitat. The tadpole is aquatic, and as an adult, the frog now has reabsorbed its tail, and it has a terrestrial habitat. So lots of things can change during this period of metamorphosis, where the larval form sometimes has an intermediate juvenile stage and then develops into the fully functioning, reproductively capable adult form. Okay, and so for instance, in humans, we don't have a very different morphology from juvenile to adult, but we obviously do have a change from reproductively incapable child and juvenile to reproductively capable adult, okay, when puberty hits. All right, so that's our metamorphosis. All right, so let's move to the next slide now and look at some pictures that are very similar to the slides that you will look at this week in lab. So there's a lot of different stages, some of which I mentioned already, but we're going to break this down a little bit further and look at examples of each of those. So first off, you got to have an unfertilized ovum, right? This is before fertilization takes place. It's haploid, single cell, and you can see that right here in this picture here, single cell. And the way to distinguish this from a fertilized single cell is that you can make out a nucleus and a nucleolus, okay? You can see those pretty readily in the unfertilized ovum, okay? And then this cell actually secretes chemicals which drive the movement of sperm in order to fertilize it. So it's a very interesting, delicate, complex, sophisticated process that actually promotes then fertilization. So this is called chemotaxis, where there's a chemical emitted, and that chemical actually draws sperm of the same species. So if this were sea star, which these are sea star cells, they emit chemicals that draw sea star sperm. If you think about this, a lot of organisms fertilize external to the body in an outdoor habitat, so in a lake, a stream, river, what have you. So if the chemicals that were emitted would draw any old sperm present in that habitat, you could get some very strange fertilization. However, there's a lot of very interesting checkpoints in place. So one of these is that the chemicals emitted attract sperm of the same species to that egg. That's just one of several. We will now move to looking at our fertilized egg. So once that takes place, sperm meets egg, you're going to see a cell that's still single-celled appearance, but now you cannot any longer see that nucleus or nucleolus, but you do see this little thin layer around the cell, and that's called the fertilization membrane, okay? And so once fertilization takes place, there's other really interesting checkpoints that take place too. make sure that the sperm that meets the egg is in fact of the same species. It won't allow in sperm that is of a different species. And also it has to prohibit, there are processes in place that prohibit a second sperm, even of the same species, from entering that. Because say you had that second sperm try and successfully penetrate the egg, then what you'd have is a triploid organism. You would not have proper then mitotic division, and so there'd obviously be some morphological and developmental problems with that organism. So there's changes that take place once fertilization happens by the first sperm, which prohibit the second sperm from meeting the egg, and this is called polyspermy. So it prohibits polyspermy, second sperm meeting. And then there's also changes that take place which lead to the next few stages, the cleavage and development, because now that that egg is fertilized, it needs to start dividing, right? It needs to divide to form 2 cells, 4 cells, 8 cells, and so forth, okay? So there's a lot of changes in gene expression that initiate this process. Okay, so from this 1 cell, you're gonna get 2 cell, 4 cell, 8 cell stage. So this is an example right here of the 8 cell stage. From there, again, you're getting 16, 32, more and more divisions, which result in different appearances in our developing blastula. So the blastomeres are the smaller cells that result from each of these mitotic divisions of the single fertilized cells. So if you look at my picture here, the 8 cell stage, each of these would be called a blastomere, but the entire thing is called a morula at this stage. So when you're about the 8 cell, maybe even 16 cell stage, it kind of looks like a raspberry or a blackberry, and this is called the morula stage, okay? And now as that continues to divide and increase the number of cells, then you're going to move to the blastula stage. So here's an example, here's another example. And this is characterized by the fact that there's now a hollow center to this ball of cells. So if you notice, the 8 cell stage just kind of looks like a raspberry, but there's no space in the middle. Whereas when you move into the blastula stage, you can see there's a hollow space, okay, here as well. And this center space is called the blastocele, and it has fluid that is contained within there. And then all of the layers of cells are on the outside, okay? And there's a difference in the sides of the blastula. So this is more evident in the amphibian than it is in the sea star, but there is a polarity to the blastula. So there's going to be a vegetal and an animal pole, we say, vegetal hemisphere, animal hemisphere. And this is characterized by the fact that in the animal hemisphere, there's a thinner cytoplasm to the cells. So cleavage happens more readily. So division of those cells happens quicker and more easily because of that thinner cytoplasm that characterizes the cells in the animal hemisphere. Whereas in the vegetal hemisphere, you have a lot more yolk, thicker cytoplasm, and this results in a slower cell division because the cleavage furrow cannot be formed as quickly through this thicker consistency in order to divide the cell into 2. So there's slower and less cell division in the vegetal pole of our blastula than there is in the animal pole of the blastula. Okay, and that's shown here. Okay, so this is an enlarged picture of a blastula stage where you've got more cell division in the animal pole, less cell division in the vegetal pole. Again, this will become more apparent when we look at the next slide and look at the amphibian. Okay, the center is, as I said, filled with fluid, and that center space is called the blastocele. Okay, and so now after the blastula stage, we move to the gastrula stage. Okay, and this is characterized by the formation of an indent into the blastula. There's going to be like a little dimple or a pore that forms into this center space. Okay, and that little indent is called the blastopore. You'll see that here. And then as that continues to, there are cells that continue to divide and move into the center of what had been this blastocele. They extend to form a tube. And that tube, the extension of the blastopore, is called the archenteron. Okay, those are both spaces. A blastopore is like a little indent, a space, which extends into the archenteron. Okay. And so at this point, we're getting the formation of those germ layers. Layers of cells, they're going to move over each other to initially form the digestive tract. And that's what's forming here, the blastopore into the archenteron. That's going to be the digestive tract of the organism. Some organisms, that first indent will become the mouth. Other organisms, that first indent will become the anus. But both ends, obviously, will be forming the beginning and end of that digestive system. Okay. All right. So I'll just mention also the blastopore, the indent in the center, is from the vegetal pole, and the archenteron is the space that continues from that blastopore. I also have labeled here that we're starting to see the germ layers. The outside layer is going to be ectoderm. That's what we call the outer germ layer. The mesoderm is going to be in the middle, and the endoderm is going to be lining the digestive tract. Okay. All right. So on our next slide, we're going to look at the formation of the blastula in a little bit more detail. As I mentioned, we have a polarity that's characterized more visibly in our amphibian. Okay, so if you look at the picture, the amphibian development, the progression of amphibian development is in the left column, and the progression of the sea star or the starfish development is in the right column. But these are the different stages that you'll see in each. So first we've got fertilization. In our amphibian, you've got the nucleus and the nucleolus in the animal pole, whereas in the sea star, you can see it more anywhere within that cell. Okay, as I mentioned, the animal pole has a thinner cytoplasm, and the vegetal pole has a thicker cytoplasm. And this results in, especially in the amphibian, results in more cell division in the animal pole than in the vegetal pole. So if you look here, this top is the animal pole. That will be the top here as well, and here. And this cell division can start to become more visible when you get to the morula stage. So you see there's smaller cells here. There's going to be more numerous cells here in the animal pole too. Again, because of the easier cleavage through those animal pole cells. And then the vegetal pole will be characterized by larger, more dense cells in the lower vegetal hemisphere. Okay. And that's not as clearly seen in your sea star. You're going to get, because of this larger characteristic of the cells in the vegetal pole for our amphibian, you're going to get actually displacement of the fluid, displacement of that blastocele towards the animal hemisphere in the amphibian, versus you don't actually see that happening in the blastula of the sea star. All right, and so the same stages take place though that we talked about already. You've got the one fertilized cell dividing into 2, 4, 8, which we'd call morula, because it now looks like a blackberry, and then to the blastula. And the blastula is characterized by that space, that blastocele, which now is surrounded by all the cells. Okay. All right. After the blastula stage, we're going to move to the gastrula stage. Okay, so this slide is showing you the very beginning of gastrulation. The top panel's in sea star, the bottom panel is in amphibian. Okay, so this happens slightly differently, again, because of the changes that we saw in the 2 hemispheres on our last slide. The blastopore in our sea star is going to start forming in the vegetal pole due to the change in shape of cells on that hemisphere, on the edge of that blastula. Okay, and the change in shape for the sea star is going to look like the cells look more like a triangle, like a pie slice. Okay, and that process is called apical constriction, because on the apical side, the cells constrict, okay, and form this pie shape to them. And so that's going to result in the formation of a blastopore, that little dimple or the indent into the center of the blastula. And that is what characterizes our gastrulate stage. This is the very beginning of gastrulation, okay? And the same process takes place in our amphibian. There's just a slight difference in where it happens and the shapes of the cells. So here's our animal pole with our blastocele. Here's our vegetal pole with our thicker cells, okay? And so the formation of the blastopore happens along the midline, just sort of into the vegetal pole, just at the very base of the animal pole, more in the vegetal pole, but in the midline, as opposed to in the center of the vegetal pole in sea star, okay? So this is where apical constriction in the amphibian is going to start happening. You get the same constriction of the apical side of those cells. However, they're going to take on more like a bottle shape. So again, a constriction on one end, a thicker morphology on the other end. And again, that's going to result in the formation of the blastopore. So you're going to get that indent. And then the cells are going to continue to move and divide and pull other cells with them to enable that pore to develop into the archenteron, to extend into the center and develop that tube, which will ultimately become our digestive tract, okay? All right, so let's move to the next slide and look at this in sea star. So this is a continuation of gastrulation, but we're specifically looking at sea star on this slide, okay? So you can see we already looked at these guys. This is the apical constriction, where you're going to get this archenteron starting to form. Now the pictures wrap around this way. So from here you go down to the right bottom picture, and then the blastopore is going to continue, like I said, to extend into the archenteron, and you're going to get to the point in the mid-gastrulate stage where the archenteron has like a bulb on the end of it. Okay, and at this point, you can distinguish between the cells that will be the endoderm, mesoderm, and ectoderm layers, the different germ layers. So again, the endoderm is going to be lining, the cells lining this digestive tract. The cells on the little knob on the terminal end of the archenteron are going to bud off eventually. Here, you see that. They're going to form little masses, which will be the mesoderm layer. Okay, that's going to be in the middle of the endoderm and the ectoderm. Okay, and of course the ectoderm is going to be on the outside. So these germ layers, there are some simple organisms where there's only two. You just have endoderm and ectoderm. But most animals that we're familiar with are triploblastic, meaning there's 3 different germ layers. The endoderm, as I mentioned, is going to line our digestive tract. The outside of the digestive tract, as well as muscles, skeletal elements, and organs, are going to be mesodermal layers. And then the ectoderm is your epidermis and your nervous system will all be developed from the ectoderm germ layer. Okay. All right, so let's move to our next slide. This is now the larval stage of our sea star. So we had just looked at our gastrula development. Okay, so that was the gastrula in sea star. Now we're going to move to the next slide where you're going to have the larval sea star. And as you can see, as I mentioned earlier, we have a bilateral symmetry. So if you cut this down the midline, the left and right of our sea star are currently symmetric. Okay, so this is before it has developed obviously into an adult where it's going to exhibit radial symmetry. Okay, so but at this point you can make out the very beginnings of the digestive tract from mouth to anus, the entire way through. So we're going to start right here where you can see the mouth. There's an opening here, which leads to the esophagus, which is a tube that connects the mouth to the stomach. Okay, so this is the stomach. And then if you can see, there's another tube here. This is the intestine, which connects the stomach to the anus. Again, so we've got mouth to anus. The entire digestive tract has formed, and this is characteristic of the larval stage for sea star. Okay, and now it will go on to further develop additional organ systems. Of course, it needs to have a respiratory system, a circulatory system, reproductive system. All of that is going to take place in organogenesis. Okay. So let's move to our next slide. Now we're kind of backing up a little bit and looking at gastrulation again, but this time in the amphibian. Okay, so we had looked at that in sea star here. Okay, and now we're going to skip to this slide here. This is amphibian gastrulation. Okay, so we looked at the blastula. We talked about apical constriction, where you're going to get now this these bottle-shaped cells that are causing that indent, the blastopore, to form. And then these cells are going to continue to move, see the arrow here, move inward. And at the same time, you're getting cells on the outside moving around to surround what had been the vegetal pole there, okay, the vegetal pole cells. So this is the formation of that blastopore, the first indent, is the early gastrula stage. Then we're going to move down to a mid-gastrula stage where you've got continuation of the archenteron. And as I said, you've got these layers of cells on the outside, the ectoderm, that are now moving around and surrounding the entire outside of the organism. So that's how the ectoderm, our epidermis in amphibian, develops. Okay, so it's going to move around here. And simultaneously, you've got division and movement of the cells from this dorsal lip moving inside, okay, to form the mesoderm and the endoderm. Okay, so again, the roles of the germ layers are the same as we looked at in sea star. So the endoderm germ layer is going to line our digestive tract. The mesoderm is going to result in muscles and skeletal elements as cell differentiation continues to take place. And then ectoderm is going to be our epidermis and our nervous tissue. Okay. All right, so, I just included a link here. If you'd like to click on this, I found a helpful video just showing in 3D models, showing the development of this process, gastrulation, specifically in amphibians. So if you wanted to take a click on that, I think the video is only like 2 minutes long, maybe 3. But I found it helpful, so I'd suggest you take a quick look at that. Okay. All right. And so after gastrulation, we've got further development. We've got organogenesis and neurulation, okay? So neurulation is, of course, the development of our nervous system. And this is going to be formed from the ectoderm tissue. And it's going to give rise to all the different structures of the brain and spinal cord, okay? So if you look here, I have listed several different terms. And I'll just mention that cephalos refers to the head, okay, cephalos. So if you see anything cephalo, that's going to refer to head brain structures, okay? So teal and diencephalon and diencephalon are going to be the foremost regions of the brain. And they're shown here. Number 1 and number 2 are teal and diencephalon, respectively. And then, so this is the dorsal side of our amphibian. This is the ventral side. Okay, this is going to be the head, and this is going to be, of course, be the tail. Okay, just to orient you. All right, so this is the brain structures. Again, everything's forming on the dorsal side. Okay, you've got the brain. Right now, it's taking up a lot of the space in the head. But again, the spinal cord is going to extend down the dorsal side, same as ours is. Okay, so you've got, again, telencephalon, diencephalon are the foremost parts of the nervous system. Following that is the mesencephalon, then the rhombencephalon, and then the spinal cord, okay, extending down towards the tail. They are most dorsal, then just ventral to the spinal cord and parts of the rhombencephalon, you're going to find this tissue here that is a supportive structure. It's got like almost a fragmented like appearance. If you notice, there's like a little bit of granulation shown here in number 6. This is the notochord. This is a support structure for the developing nervous system and the animal in general. Okay, and then further ventral to that, we've got the digestive tract. Okay, so at this point in development, there's no mouth yet. However, there is an anus that was, formed from that first blastopore, right? And so we've got the anus, the hindgut, the midgut, the foregut, and then just anterior or rostral to that is our pharynx. Okay, number 7. There will obviously be a mouth eventually. Okay, so look for, I always recommend looking for the nervous system tissues first and that notochord, and then looking for the structures of the digestive system. You can identify a lot just by looking, a lot of anatomy, just by looking for the nervous system and the digestive system. And then lastly, you want to look for other tissues and organs that are going to be sandwiched in between or most ventral. Okay, so we've got the heart in the pericardial cavity right here. So number 13 is your cavity, the space that's going to surround the heart, number 14, okay? And then number 12 here is the yolk. That's what we had seen in the vegetal pole when we were looking at the gastrulation process. Okay, so that's going to be what's left of that. And of course, the entire organism has been surrounded now by ectoderm tissue forming the epithelium, okay? All right, so you are going to be looking in lab this week at slides that look similar to this. This is obviously a drawing, but you'll be looking at slides that look similar to this and similar to this next picture here. I've kind of just included this a couple times to show you a few different pictures that resemble what you should look at. Okay, so these are sagittal cuts through the organism. You're going to be looking at frog embryos sagittally as well as in cross section. Okay, so these are cross sections. So if you look at, this is the sagittal picture that I'd shown you. If you look at the lines, the red lines that I'm showing through different points from the head to the tail, they are going to correspond roughly to the pictures that I have here in cross section. So if you were to cut the organism down along line number 1 here and then look at the cut side, this is what you should expect to see. Okay, so you want to be able to identify different anatomy in both sagittal and cross section. Okay, so this is the foremost cut, foremost picture that I've included, showing you the diencephalon, which is this nervous system structure here, okay, and the eye, which includes the optic cup and the lens. Below that is the pharynx, and then these little glands here, these little projections here, I guess I should say, are oral suckers. Okay, they allow the organism to adhere to a surface and enable feeding. All right, moving to number 2, we're moving further caudally down towards the tail, right, of the organism. So we're looking at sort of a slice around this point where I've drawn the red line. And again, if you were to look at that cut section, you'd see something like this. This is the dorsal side. This is the ventral side. So if you remember, all the nervous system structures are going to be towards the dorsal side. Okay, you saw the diencephalon. We say that if you see the eye, then you can identify that you're probably at the diencephalon, eye, okay? Diencephalon is at the level of the eye. And then the rhombencephalon is characterized by this very thin layer of cells on the dorsal side and much thicker masses of cells on the ventral side. And I'll also mention that these spaces here, you've got spaces at all the different points in the nervous system that the actual tissue surrounds, okay? So this would be, for instance, a ventricle of the brain. In the brain, human brain, we've got ventricles, spaces, where cerebrospinal fluid travels, and that continues, of course, down all the way through the spinal cord. Okay, so this space, this clear or white area is a space, and the nervous tissue is actually what surrounds that. Okay, the purple in this picture. All right, so you've got a thick lower wall or ventral wall to the rhomencephalon. And again, the rhomencephalon is characterized by a really thin dorsal wall. Okay. And now in the position where you had seen your optic cups in slice number 1, now you're seeing a hollow space. This is the hearing organ for our frog. This is called the otic capsule. Okay, this is the hearing organ. And then you're starting at the level of the rhomencephalon to see that notochord that I mentioned, the support structure. It's going to be in the midline, just ventral to your nervous system structures. Okay. And then the pharynx has thinned out. The pharynx takes on various types of appearances, various morphologies, depending on where you are looking, which cut you're in, head to neck region. So it can sometimes look very thin like this. Sometimes it's a larger open cavity, depending on which slice, which position you're at in the organism. Okay, but this is the pharynx. At this point, it's thinned out right here. It's going to lead to the gills on either side. Okay, so that's one way to recognize that that's the pharynx. Okay, and then most ventrally, we're looking at the pericardial cavity and the heart with the atrium and ventricle. Okay, all right, so let's move to picture number 3 here, where again, you're seeing some rhomencephalon. You can again tell that that is the thin wall and the thick, more ventral wall of the rhomencephalon. The notochord, it got a little bit displaced, it looks like here. It should be more on the midline, but sometimes when slides are prepared, the organism gets a little squished or is cut kind of funny. So, but that's the notochord. It's going to be round. It's going to have like a fragmented appearance to the inside of it, okay? And then you've got what we call pronephritic ducts or tubules. So there's little circles here, all these little circles. These are what will become the kidney structures of the frog, okay? And then in the most ventral region, you've got your gut area, including a liver. The space around the gut is called a coelom, and you might have seen that up here too, pericardial coelom or cavity is just the space, okay, around those structures. Moving to our 4th slide here, you now move from the rhomencephalon, which is part of the brain, to the spinal cord, okay, as we extend more towards the tail. Okay, so the spinal cord is going to be much smaller and rounder in appearance, and if you notice, you've lost that flat wall, okay, on the very dorsal side of the nervous system structures is now much rounder, okay. All right, so here's our spinal cord. Again, you still got the notochord, just ventral to that. And then you've got a yolky endoderm, which is going to be present most ventrally. Okay, still got some of those pronephritic ducts and the space called a coelom around the endoderm. Okay, and that would be corresponding to line number 4 here. All right, I think that really has walked us through most of what you'll be looking at in the slides this week, okay? Now we are going to just cover, again, reiterate that after this point, a lot of organisms go through a metamorphosis from juvenile to adult form. So for instance, in sea star, we had mentioned that you go through stages where that bilateral larval form is converted to a radially symmetric adult form, okay? And for frog, as we mentioned, we go through the tadpole stage, the tail is reabsorbed, you've got 4 weight supporting limbs in the frog, and of course, a reproductively capable adult, okay? And for humans, Same thing, we don't have a lot of changes in our body plan, but we obviously go through a change from juvenile to reproductively capable adult. I just included a picture of a caterpillar to butterfly because that's what people often think of when they think of metamorphosis. That's just another example, right? Major change in body plan, which many organisms go through, although we don't. Okay. All right, so that wraps up the material for the pre-lab for animal development. Any questions? As always, you can shoot me an email, cfry@lvc.edu, and I will see you shortly. Have a good day.